Hello guys, this is Vineet and welcome to the molecular biology series, chapter number 3, discovery of the heritable material. So today I'll be discussing about the discovery and history of the genetic material, that is the DNA. So it was all started by the Mendel and also by the Mischer same year. Uh, Mendel was working on the hereditary patterns or the patterns of heredity in the pea plants. Okay, at the same time Mischer was also working on the white blood cells to uh, know what was the transforming principle or the what was the material that was transforming from one generation to another generation. In his laboratory Mischer was working on the white blood cells, and when he transformed these white blood cells into weak basic solution, this weak basic solution caused this white blood cell to swell up and undergo degradation of cell nuclei. And this cell nuclei, when it got exposed out, it released some kind of white color material, and he was able to extract that white colored material, and he found that the same material was also formed in the yeast cells, yeast nuclear cells. Okay, and he also concluded that these white colored materials contains the carbon, hydrogen, oxygen, nitrogen, and phosphorus, and he named this material as the nuclein. Okay, so first the curiosity about the genetic material was started by this Mischer. Okay, and then in 1884 the Oscar Hedwig, so he concluded that nuclein is responsible for the transfer of the inheritance based on the Mendel's experiment only. And in 1909 scientists from the Rockefeller Institute, so they conclude that they concluded that ribose is the sugar in the nucleic acids. Okay, so not only the ribose is present in the nucleic acids, at the same time, no, the American biochemist Phoebus Levin, so he analyzed the chemical composition of the nucleic acids, and he said that there were two of such. Okay, so two types of nucleic acids were there. So one is the RNA and DNA, and he said that it contains a nitrogenous base, pentose sugar, and also the phosphoric acid groups. Okay, and he also said that the sugar which is present in the DNA doesn't contains oxygen group, oxygen atom, carbon 2 position. Okay, so that means a deoxyribose sugar is present in the DNA. Okay, and that is why it is named as the deoxyribonucleic acid. Okay, and at the same time in 1944 at Rockefeller Institute, okay, so Avery, McLeod, and McCarthy was continuing the experiments of the Griffith to find out the transforming principle, the major principle behind the transformation experiments by the Griffith experiments. Okay, in the Griffith experiments. Okay, and they have succeeded uh, finding out the DNA is the only thing which is responsible for the transformation principle. And then in the middle of the 20th century, Watson and Crick, they show the structure of the DNA. They have actually constructed a double helical structure of the DNA, and they also gave the actual diameters and the distance between the two bases and all these things. Okay, entire properties of the structure of the DNA was given by the Francis and Crick. Okay, and they allowed most of the famous scientists to understand about the genetic material. Okay, and how does it transfer from the one organism to another organism. Okay, so this was about the basic history about the discovery of the material. And as I said you, it was all started by the Mendel. So when he was working on the pre plants, he concluded that there was some principle transfer from the uh, parent plants to progenies. Okay, and he also did a lot of experiments on the pre plants and gave three laws of inheritance. Okay, one is the law of dominance, and one is the law of segregation, and one is the law of independent assortment. Okay, however, the law of dominance is not considered as a major head genetic rule in today's world, because this law of dominance is overruled by the nature in a lot of things. Okay, so most of the recessive genes or recessive alleles, they also dominate the dominant alleles. Okay, so because of that, law of dominance was not acceptable. But the law of segregation and the law of independent assortment were the major things which were proposed by the Mendel. Before the experiments on the pre plants, he also worked on the honeybees, uh, like 3 to 4 years. Okay, and he his results weren't that accurate, because the honeybees contain the haplodiploid system in them. So the result was not accurate, or the results were not going according to him. And again he came back to the he came to the he conducted the experiments on the mice, the transformation experiments on the mice. Okay. And lot of Christian communities or the priest communities at that time, they have opposed this Mendel experiment, because those experiments was against the ethics. Okay. And then he finally came uh, with the pre-plants. Okay. He conducted the experiments on the pre-plants, and he proposed the laws of genetics. Okay. And then the Misher, he identified the nuclein as the material which is present in the cell nuclei. And then the Sutton and Boveri, they proposed the chromosome theory of hereditary. And then the Morgan came, he coined the term linkage and explained about the linkage. Okay. And then the Mueller, he used the X-rays to induce the mutations in the genes. Okay. And then Griffith, he started the transformation experiments. He was the major person to build the curiosity to know what is the transformation principle, that was transforming from one parent to another progeny. And then the McClintock, she was working on the transposable materials. So she said that there were two types of transposable materials. One is the cut paste transposons and the copy paste transposons. So, and these were responsible for increasing the genetic material quantity, and they were able to cut the DNA sequences, and they were able to paste themselves at that cut regions and replicate themselves. Okay. So these were the major findings of the McClintock, and she worked on the corn plants. Okay. And the Beadle and Tatum, they gave the one gene, one enzyme hypothesis. That means, only one gene codes for, one specific enzyme. That was also not true. This was also overruled by a lot of mRNAs in nature. And then the Avery, McLeod and McCarthy, who continued the Griffith experiments. Okay. So they concluded that, the DNA was the transforming principle, which is responsible for the hereditary. And then the Hershey and Chase, they used the radioactive labeling to prove that, DNA is the only material responsible for the hereditary. So all the experiments till Hershey and Chase were made to prove that the DNA is a genetic material. Okay. And it was the transforming principle. Okay. And then the Watson and Crick, okay, so they gave the structure of the DNA molecule. And then the Jacob and Monod proposed that there were the mRNAs existed in the cells. Okay. So, and these mRNAs were able to translate into proteins. Okay. And then the Human Genome Project has begun in 1990s, and it was completed around 2003. Okay. First genome project, and it has revealed a lot of unique facts about the DNA. So one major fact is 98% of the DNA is not needed, which is uncoded, while only 2% of the DNA is useful one. Okay. So this is about the basic history and the discovery of the heritable material. So one major experiment which was carried out by the Griffith is the transformation experiments. Okay. So he took two strains of the streptococcus pneumonia. So one is the virulent strain and the one is non-virulent strain, and he named this virulent strain as the S strain, and he named the non-virulent strain as the R strain. Okay, so you can also call that as the smooth strain and the rough strain. Okay, smooth strain contains a polysaccharide coat, while R strain doesn't contain any kind of polysaccharide coat. So its cell is exposed. Okay, its cell is exposed to a lot of degrading enzymes. Okay, so whenever you infect this S strain bacteria to the mice, the smooth strain was able to replicate and cause the pneumonia to the mice, and because of that thing the mice dies. Okay. And when you infect this R strain to the mice, okay, so the cells contains a lot of degrading enzymes, and these degrading enzymes were able to break this R strain bacteria, R strain, and the mice mice cells, by degrading those kind of the R strain, so they were able to live. Okay, so there was no death seen in the case of R strain. Okay, because it doesn't contain any kind of polysaccharide coat to protect the R strain molecule. Okay. And when he heat killed the smooth strain molecule, smooth strain, okay, so when he subjected to heat treatment, okay, so this heat denatured the polysaccharide coat, okay, and it became the R strain type thing, and when he injected the heat kill strain to the mice, okay, and because of polysaccharide it was able to grow. Now the polysaccharide coat is not there, so the mice cells has the enzymes. Now this enzyme can degrade the heat kill strain and the mice can able to live, okay. And when he mix this strain with R strain, okay, heat kill strain, and he mix it with the normal rough strain bacteria, and when he injected both of them to the mice, okay, by his observation mice died, okay. So the reason behind is there was some kind of transforming principle or some kind of transforming material that was getting transferred from the smooth strain to R strain, so that the R strain could synthesize a polysaccharide coat around itself like this and able to infect the mice or become as a pathogenic strain, okay. So because of that reason the mice has died, okay, and he found that their lungs contain live pathogenic strain of the S. pneumoniae. So that means something has transferred from the heat kill strain to R strain, okay. What was that something? So this, before his conclusion, the Griffith died, and his experiments were carried out by the Avery, MacLeod and the McCarthy. So he extracted the DNA, proteins and RNA from the Streptococcus pneumoniae strains, okay, and he took 4 test tubes, okay. So 4 test tubes contain the equal amounts of the proteins, DNA and RNA solution, okay. To the first test tube, they took this first test tube as the control, this one, okay. That means the no enzymes were added to the to this test tube, and to the second tube, this tube, so only proteases were added, okay. So proteases were meant to degrade the protein molecules, so that means they can eliminate the protein molecules, okay. And then for the third test tube, so they have added the ribonucleases, which are also called as the RNases. So these RNases were able to degrade the RNA molecules, this red color molecules, okay. And to the to the fourth test tube, so they were able to degrade the DNA, okay. So deoxyribonucleases cleave the DNA strands, okay. And when the test tube contains the deoxyribonucleases, so the DNA which is present in the test tube will get degraded, okay. And they have transferred the mixture which is present in the 4 different test tubes into bacteria. So they have actually added the R strain Streptococcus pneumoniae cells to all the test tubes, okay. So, and they have observed the experiment, okay. So what was happening, and they finally concluded that the R cells which were added to the test tube which contains the deoxyribonucleases, okay, in that test tube his cells were absent. That means transformation did not occur because the deoxyribonucleases have cleaved the DNA of the smooth strain bacteria, and the transformation did not occur. So they conclude that DNA was the only material that will be transforming from the S strain bacteria to R strain bacteria, okay. And in the cells which in the test tube which were in the test tube where the ribonucleases were added, okay, so they only cleave the RNA molecules, since the RNA molecules were not the transformation material, okay. So the S cells were able to, R cells were able to transform into S cells, okay. So the rough cells were able to transform into smooth strains, okay, and able to infect the mice and unable to kill the mice, okay. And since the proteases cleave the proteins only, and the proteins were not the actual substances that transforms the material thing, the actual, not the genetic material thing, okay, proteins. So these proteins were also degraded, and the R cells can able to convert into those pathogenic strains, the smooth strains, okay, and they were able to infect the mice, and the mice also died, okay. So in all cases the mice has died, but in the case of in all the cases mice was alive, but in the case of in all cases mice was dead, but in the case of this deoxyribonucleases, okay, so the smooth cells, they weren't able to convert the R cells into smooth strains because there was no DNA, and the mice was alive, okay. So in this case only mice growth was seen, okay, because there was no DNA, because the deoxyribonucleases cleaved the DNA, okay. So they concluded that only DNA was a transforming principle, because the deoxyribonuclease acts only on the DNA, and the DNA was cleaved in the 4th test tube. So that was their experiments, okay. So in order to prove the actual uh, information and how to prove the actual transformation principle, about the DNA. So this Hershey and Chase label the bacteriophage, the bacteriophage which infects the bacteria. So it is a virus which infects the bacteria. They labeled the DNA with phosphorus 32 because the DNA contains the proteins. Sorry, DNA contains the phosphorus, and they labeled the protein code with the S35 because the proteins contain the cysteine amino acid which are made up of the sulfur. Okay, and they were able to transfect the bacteria, and they cultured these infected bacteria. Okay, by their observation, bacteria infected with the phage containing the phosphorus 32 labeled DNA produce a P32 labeled phage. Okay, that means the DNA was transferred from the parent phage to the progeny phage. Okay, and the bacteria which was infected with the S labeled phage, the protein code labeled sulfur labeled protein code phage. Okay, so it wasn't able to produce the labeled phages. Okay, only the unlabeled phages were produced. Okay, so that means only DNA is transferred from the infected phage, or sorry, labeled phage to progenies. Okay, not the protein code. Okay, so this experiment has concluded that DNA is only the transforming principle that transfer from the parent to progeny. Okay, and then Biddle and Tatum came. So they were working on the fungal spores. So what they did is they said that the spores of the fungal species, they have all the enzymes to use the vital nutrients and grow on those nutrient media. Okay, but when we mutate the some genes of that fungal spores, they cannot grow on the specific nutrient medias. Okay, because the genes were mutated, and the genes were not functional, and the genes were not able to code specific enzymes. Okay, so suppose conclude that these fungal spore strains, okay, spore A, okay, so they were subjected to the X-rays. Okay, and these X-rays have mutated one type of gene. Okay, so this gene was responsible for the production of an enzyme. So this enzyme makes the convert the nutrients of this nutrient media A, okay, into vital nutrients. Okay, and it is responsible for the growth of these fungal spores. Okay, so when you mutate this gene, the gene which is responsible for the growth of an enzyme which converts the nutrients into useful substances, if you mutate this gene, the gene won't be able to produce any enzyme. Okay, now the fungal spores cannot survive on this nutrient A media. Okay, the same thing did with the second type of fungal spore. They have mutated different type of gene, okay, which were responsible for the production of an enzyme. So that enzyme makes this fungal spores to grow on the nutrient B, different type of nutrient. Okay, the same thing has observed, and they have concluded that even those fungal spores are same, okay, but the genes which are present in them codes for specific enzymes. Okay, but this concept is also overruled by the nature, because a lot of genes when they get transcripted into the mRNA, so this mRNA gets spliced into different components, and a different component codes for different types of proteins. Okay, so this concept was also overruled by the nature, and this was about the history and the discovery of the heritable material. So in the next class we'll discuss about the structure, properties and functions of the DNA.